Hello, everyone. I would like to start my presentation. So I'm going to switch gear a little bit from protein to actual the minimal cell lipidome and actually the remodeling machinery. So my work focuses on the biological membrane. And for biological membrane, like John mentioned, even the mammalian cell or minimal cell, they have to perform a certain types of function. So they have to be semi-permeable, they have to be robust enough for outside stress, and also they have to be performing as an organizational platform for multiple enzymatic process. And in order to do this, they use lipids and actually a lot of different types of lipid. So this create which lipid are there? So for mammalian cell and a lot of our minimal cells, they need cholesterol. And most of the complexity comes in with the other types of membrane lipid, where you have one head group and 2 or 4 different acyl chain. So the head group can have different structure, and the acyl chain can also be different too. It could be longer, shorter, with a double bond or not. All of this combined with each other can create a myriad of different types of lipid. And all of them have different function. That's why you have a very complex membrane. And you also need to know how to use the membrane, which makes the regulation machinery very difficult to understand. That's why we look to the minimal cell in the hope of looking at a more simpler, a more straightforward remodeling system. And in this crowd, I probably don't need to introduce the JCVI-syn3B we have here. And earlier this year, we have a manuscript on how the lipidomes of JCVI-syn3B changes when you apply different temperature on it. And I would also look into the lipidome of Mesoplasma florum, which is a closely related species. This one has 720 genes, which is more than JCVI-syn3, but we have reason to believe that they also are, but they are also very simple, and we want to compare them to see if they are actually using the same strategy into how to manage the lipidome. So what do we actually know about the remodeling capacity of these two organisms? If you look at the annotated pathway for JCVI-syn3 and Mesoplasma, they both can produce 2 types of lipid head group, which is PG and cardiolipin. And from our lab's observation, they can also produce DAG or DAG. So that makes 3 types of lipid. However, all of the other types of lipid, like cholesterol, sphingomyelin, PC, PE, they cannot produce themselves at all. They can take it up from the media and put it in their membrane, but they cannot synthesize the head group themselves. Okay, so if they have a very similar enzymatic capacity, will they have the same membrane? So we try to look into that by feeding them both serum, so very complex lipid profile, and look into the distribution of lipid over different class. And this is how the lipid distribution looks like. So from the first glance, you can already see they have different colors, which denote different lipid class. And I can put them like this. Maybe it's easier for you to see that even though they have some similarity, like they both use sphingomyelin or SM here and cholesterol, the amount of PG is different for the 2 organisms. And surprisingly, minimal cell actually have 2 different lipid class that Mesoplasma doesn't really enrich in the membrane, which is PC and cardiolipin, while Mesoplasma have the DAG that JCVI-syn3 doesn't really use. So first shot, you already have the static picture of the 2 different systems that these 2 organisms use, even though their enzymatic background may seem similar to one another. But how are these 2 systems performing when you actually need them? So biological membrane has a function, which is to homoviscously adapt to temperature. And what does this mean? If you have just the same lipid setup of and you change the temperature, a thing called a lipid packing, which is measured here by Pro12 or Silauden general polarization, changes. And this is not good for the cell because a lot of their other proteins are optimized for a certain range of lipid packing. And once you change lipid packing, all the other things can function less efficiently. So the role of the biological membrane and the role for the organism to remodel the membrane is actually change the composition of the membrane to make sure that the spread in difference of lipid packing is less. So syn3 can do this in their growth temperature range from 30 to 37 degree, and Mesoplasma can do this in their range from 15 to 38 degree as well. So both of them can viscously adapt, which means they can shuffle their lipidome. And how does it look like? So if you look at it in bulk, so here you see plot which denote the abundance of lipidome change in mole% normalized by temperature. For both syn3 and Mesoplasma, they are roughly at around 3 mole% per degree, which is fascinating considering that these two organisms has very different growth temperature range. And if you take an even closer look at different lipid class, so in this plot on the y-axis, you can see the class abundance, and in the x-axis, you can see different classes of lipid. And first, I want to show you, and also the opacity is the growth temperature. So more opaque is higher growth temperature. And for the imported lipids, you can see that syn3 remodeled changes the class abundance very significantly and dynamically. From cholesterol, sphingomyelin, PC, they all varies. While for Mesoplasma, they don't change that much. But it is completely different for the lipid that they synthesize themselves. While syn3 only really changes the class abundance for cardiolipin, Mesoplasma very dynamically adapt their class abundance of PG and DAC according to the growth temperature. And if you look if you even take a closer look at how the composition of this lipid looks like for the two organisms. So first, I will show you syn3. In this plot, you can see on the X and Y axis is the average double bond and average length per azo chain of the two different lipid class. And again, the opacity is denoting the growth temperature. You see that from more transparent to more opaque, PG and cardiolipin seems to be following two different patterns. This is called head group specific lipid remodeling. And this is a common pattern seen in multiple organisms. We see it in Methylobacterium, you see it in E. coli, in yeast, and apparently here we also see it in syn3. But for Mesoplasma, if you look at the lipid that they produce themselves, which is PG and DAC, you can see that from transparent to more opaque, so increasing in temperature, the pattern for azo chain double bond and length actually match quite well between the two different lipid class, which means they do not have head group specific remodeling. This means that Mesoplasma perhaps have an even simpler lipid remodeling mechanism comparing to syn3, even though their genome size is, of course, larger. So this is a little snapshot of what we are working with. What I want you to take home from this presentation is first, the even minimal membrane can retain a certain amount of complexity. And even though Mesoplasma has a larger genome size, they might have a simpler lipid remodeling machinery that is worth looking into. And this is even simple, very robust, since you look at the temperature range that they can adapt to is wider than even the growth temperature range of syn3. And in the future, I want to take a closer look into the two different systems, the cardiolipin, which is what syn3 uses, and DAC, which is what Mesoplasma uses more. So the thing that is in common with these two types of lipid is actually both of them have negative curvature. This type of lipid is very important for determining the cell shape and also cell division. So I would like to use SYN3 and Mesoplasma in a more or less comparative biology case to see what are the different roles of this two membrane system and is there any benefit to use one to or the other. And if there is actually a benefit to using the DAC system over cardiolipin, maybe we could even think about introducing Mesoplasma system into SYN3 and hopefully increase its robustness. Okay, so that's it for my talk. Thank you all for listening, and I hope to take your questions.
That's terrific. I'm just filled with questions.
Lovely.
The first thing I want to ask, we know that genome transplantation works better if the recipient cells are grown at 30 degrees than if they are grown at 37 degrees.
Okay.
So hearing that, from a lipid perspective, any thoughts on that?
So from the lipid perspective, I think it also for us makes sense to have more context because for SYN3, for example, you see that 30 degree is already in the lower end of the lipid of their growth range. But for Mesoplasma, it is actually just near its optimal temperature. So I'm not, so their lipidome, if I can rewind the slides a little bit and maybe make up more of a point here. If you look at Mesoplasma, when you look at the two ends, when they adapt to cold and when they adapt to heat, actually they have to remodel the membrane more. So the membrane seems to be more stressed. So if you say that the genome transplantation works better at 30 degree, maybe in this case for SYN3, the membrane is more stressed and thus maybe compromise the membrane integrity and makes it easier for genome transplantation. I would not necessarily say that Mesoplasma might follow the same path.
Okay. And then the other question, so we know that the wild type Mycoplasma mycoides has to live in the upper respiratory tract of a goat at, say, 30 degrees, and the lower respiratory system at 39 degrees. And we also, there are likely a number of transcriptional regulatory differences between 30 and 39 degree growth. Does it make sense that you have one set of, or one kind of lipid diversity at 30 for the upper respiratory system and something different at 39, where in the lower respiratory system, whereas Mesoplasma, it's going to be in plants, and plants are going to have the same, the same temperature as the environment that they're grown in, but it's likely to be that the Mesoplasma is in the same parts of the plant either way.
Yes, and I think that might be the contributing factor for why Mesoplasma actually can adapt to such a wider range of temperature, is because they are not their hosts might not be very good at thermally adapt and retain the temperature at all times. So for for mycoides, they live in mammalian, which can retain temperature, and they usually don't change that much. But Mesoplasma, it could some people say that it comes from plants, some people say that they are from insect, but either way, they are not very caudal in terms of being able to live in a thermostat all the time. So maybe that's why we have machinery that is more flexible in terms of temperature adaptation. But here, what we find fascinating is that even though the task is considerably more difficult, the machinery might be even simpler, which and this pattern of the exact same average acyl chain length and double bond into different lipid class is not very commonly seen in any other organism. Head group specific remodeling is usually more the trend here. So we find it fascinating to see such a simple system working so well.
Thank you. Other questions?
Yeah, I have one. Kim Wise again. I have a question then. So since your remodeling in Syn3A is done through these synthesized membranes, how do you envision the regulatory system to work on that? Are there other known organisms where they figured out what the feedback loop would be to actually regulate whatever it is, if it's just transcriptional, or is there any ideas on what that might be?
So that is a very open field right now. A lot of people are working on it. We recently have a publication of the case in yeast, where they find a protein system that a protein that basically censor the membrane properties in different parts of the bilayer, and that create the feedback system to the other one. But other than that, what we see is more of a response factor but the amount of protein that actually can censor the changes in membrane properties are still undergoing to be characterized.
Mm-hmm. But they must be in the Syn3A genome, so maybe that's some of the unknown proteins. Okay.
Yes, membrane proteins are very complex, and we all also really want to look into that.
Yeah.
John, this is Prescott. Could I ask a question?
Please.
I was really interested to see the proportion of cholesterol in the membranes. I mean, I'm pretty ignorant about membrane lipids, and used to seeing, like in your little cartoon in the beginning, you showed little bitty cholesterol molecules scattered around in the lipid bilayer, and then in the pie graph, it's like it's the dominant lipid, and I really don't understand how it fits in amongst all of these other lipids. And I guess my question is, do you see any changes in the proportion, since I seem to remember that cholesterol has to do with membrane fluidity, and fluidity is something that's going to change with temperature. I wonder if the proportion of cholesterol in the membrane is changing also when you grow it at different temperatures.
Yes, absolutely. Actually, in the manuscript, in the paper that we just published, we see both, not only that cholesterol changes in when you adapt to temperature, they also changes at a different rate comparing to other membrane lipids. So in this manuscript that was that is from Natasha Safronova here, I think in the later figures, you can see that while other membrane lipid or phospholipid go through remodeling relatively slowly, cholesterol actually plunges in the beginning and then recovers. So it kind of creates a slow and fast response for the cells as well. And in terms of the actual abundance of cholesterol, yes, every time we present this data, people are amazed at how much cholesterol there are, because for Syn3 or Mesoplasma or mycoides, they are all higher than 50%. And it is almost to the maximal solubility of the cholesterol in the membrane before it crash out. So we are also still looking into why that could be, and why does the cell want their membrane to be such a critical state. So that is a very fascinating thing indeed.
Other questions?
So here's Bogumił from Western University. I was curious, do you ever actually sequence those strains that evolve to different temperatures just to see what happens there?
I think I don't remember specifically having sequencing data. We do have a set of transcriptomic data, but I'm not sure if it's temperature with a different temperature stress yet.
Okay, I'm just curious because the strain that Daniel presented, that he evolved for better transformation, we sequenced that and we actually noticed a lot of mutation. So it was adapting very quickly to different things, but it changed forever, kind of. So that was curious. And finally, obviously, I'm gonna pitch here as we are preparing the genome transplantation Lipidome that grows, it's slightly different conditions. You don't need the ABS and so on. Would you be interested in looking at that membrane of this microbe? I don't know. We can obviously talk about this later, but I would be very curious to see such a study done on that. So something to.
Yes, actually acrylic plasma is also on our radar in terms of I specifically work on sterols, and acrylic plasma is good for me because they also don't need a source of sterol. So yes, we would be very interested into looking into the membrane. But also to circle back on your point in terms of sequencing them at different temperature. I think last year during the JCVI workshop, there was also a work presenting on, I'm not sure if it's mycoides or syn3, but actually adapting them to higher temperature. So one of the things that could be done is after you adapt them to a higher temperature and you can put them back in the older temperature, how fast are the membrane changing back to the way it was, or if they can do it at all, if they already permanently changed their sequence. So those are the things that is definitely on the radar and we would like to pursue in the future.
Nguyen, the next shipment I make to your lab, we will include the strain that grows at either 43 or 44 degrees. So you can play with that.
Yes. And can you remind me if that is syn3 or mycoides?
That was syn3B.
Ah, lovely. Thank you.
And we sequenced it, but remember, over extended growth, you accumulate a lot of mutations. So it's hard to know. There are more than a dozen different changes in the genome. So it's hard to know which ones are relevant to the higher growth temperatures and which ones are just spontaneous mutations that happen because we didn't sequence 10 strains, we just sequenced one. When we do this again, we'll do a better job with this.
Yes, in either case, I think it will also be beneficial for us to see how well it readapts to the older condition. So that could already be a first level readout in terms of is anything affected at all.
So this might be something, if Yo Suzuki's lab isn't interested in doing this, this might be an experiment we can get you the cells and let you guys play with.
Yes, that would be really amazing.